So good morning, everybody. A few of you are not here yet, so they will be scolded later. But it's a pleasure today to introduce the new speakers. We start with Lynn Uhrig, who is a medical doctor specialized in anesthesia and with a PhD in cognitive neuroscience, right? Yes. And she's at the University
yes, I'm working
of Paris,
in Paris, yes.
Saclay, and also working on neurospin, doing specialized research on models of anesthesia in rat as well. In mice.
Monkeys.
And monkeys, sorry.
Monkeys, yes.
Big rats.
Yeah, big rats. Big rats. So today she will be talking to us about cerebral mechanism of general anesthesia. So I leave the word to her, to Lynn.
Okay, thank you very much for the kind introduction, and good morning to everybody. So my talk will be about the cerebral mechanisms of general anesthesia. So like Anna talked to you at the beginning, my background is that I'm an anesthesiologist, and after my medical studies I did a PhD in cognitive neuroscience, and the goal was to study cerebral mechanisms of general anesthesia because I wanted to know how it really works when you use general anesthesia in everyday life for people. So that's a small overview of my talk, with first a small introduction of mechanisms of general anesthesia, and then I will show you the monkey models we developed in the lab to study general anesthesia with using functional MRI and auditory stimulation, and also after we used fMRI and resting state to study anesthesia, and at the end I will present you a general conclusion for the mechanisms of general anesthesia and some perspectives for the future. So when we talk about anesthesia, anesthesia of course is used in everyday life for thousands of people in the world to do a surgery. But mechanisms of the anesthesia are still not completely known today, so it's used. It's quite safe when you use it in everyday life, but often you don't know really what you do with the people. You know that they will at some moment lose consciousness. They don't remember what happens during the anesthesia, but why they lose consciousness you often don't know really. What is quite well known is the molecular mechanisms of the anesthetics. So anesthetics are pharmacological agents who target specific receptors in the brain, and once they target these receptors, they induce the modulations of brain regions, and at the end you have an interference of neuronal networks, and at the end you lose consciousness. So when we talk about consciousness loss in general practice of anesthesia, it's the people become unresponsive to stimulation you present to them. So you ask them, please open your eyes, and the people don't open your eyes in clinical practice. But the anesthetics we used in daily clinical practice are pharmacological agents who have different molecular structures, like, for example, propofol or ketamine, and they act on completely different molecular targets in the brain, like propofol acts more on GABA receptors in the brain, ketamine acts more on NMDA receptors, and at the end they all alter the neurotransmission of the brain and they induce loss of consciousness. So it is quite a challenge to link the molecular studies of anesthesia with the studies of loss of consciousness using mainly fMRI, what I present you after. When we present the anesthetic agents, so you have different anesthetic agents. You can use intravenous anesthetic agents, mainly like propofol or ketamine. It's the two molecules I will talk later in my talk. And also you have volatilic anesthetic agents, like, for example, sevoflurane. The main targets, like I told you, for the molecular agents are the GABA receptors and the NMDA receptors. Propofol mainly targets the GABAergic receptors in the brain. Ketamine mainly targets NMDA receptors in the brain, and like sevoflurane, it targets both receptors in the brain, and then at the end you have consciousness loss for all these molecules. So what about propofol? So propofol mainly acts on the cerebral cortex. It targets where the GABA receptors and enhances the inhibition of the GABA receptors in the brain. So first it targets these receptors on the cortex. So you have first an inactivation of the cortex, and then in a second time you have an inactivation of the subcortical structures in the brain, and you induce loss of consciousness. This was quite well studied in humans. It was studied in patients who had surgery for Parkinson's disease. So they went under general anesthesia. These patients who had an electroencephalogram put on her head to monitor the cortical activation of the brain, but also they had this deep brain stimulation electrode to stimulate the brain, but also this electrode could record the subcortical activations. And in particular the thalamic activation of the brain. And what they could show in this paper like 10 years ago, that first when you induce a loss of consciousness, it is first that you deactivate your cortex, but only like 10 minutes later you have a deactivation of your subcortical regions. So at this point you consider that your subject is not conscious, at least clinically he's not conscious, but there are some regions of the brain that are still conscious, and so they could demonstrate that for propofol, but also for sevoflurane, it acts first on the cortex and then only acts on subcortical regions. So that's for propofol, but propofol is not the only anesthetic drug used in clinical practice. There's also like ketamine. Ketamine has a complete different mechanism of propofol, and it targets NMDA receptors, so glutamatic receptors in the brain, and there is an inhibition of these receptors in the brain. And ketamine only acts on the cortex, like the whole cortical regions, like also the limbic system, the hippocampus, where it's inactivated, but the subcortical regions, in particular the thalamus, is still active under ketamine anesthesia. And when you look on an EEG that it's acquired under ketamine anesthesia, you see that you have an EEG that's very active, so it's not slowing down the activity of the brain. It's only propofol that's slowing down the activity of the brain, but at the end, all these people are unconscious if you use propofol or ketamine. So at least at this time, you don't really understand why both could induce unconsciousness with complete different mechanisms of the brain. One other anesthetic agent, I will not talk very much about these agents, is dexmedetomidine, but I think you have to know that this molecule exists because if you use it in the clinical practice, it will induce like a sleep-like state that's very similar to sleep, but it acts only on subcortical regions and not at all on the cortex, at least what is described in the literature. So how can we study the anesthetic agents when we doesn't want only to study the molecular actions of these anesthetics? So what we know that anesthesia gives a very low vigilance state, so the people or the animals aren't awake at all when we use anesthetics. And what is commonly described for anesthetics, that also it decreases the awareness state. So with the information processing that normally it's not possible in the brain when you use anesthetics. So what was described in the past is that like general anesthesia would give a shutdown of the brain, but I will show you that it's not completely true. So first, I will describe like the more cellular and receptor mechanisms of the anesthetics, but now I will come to the function of the brain and anesthesia. So how could we study the function of anesthesia in the brain? The main technique I will show you that we use in the lab is functional MRI. We used functional MRI because it was like a good compromise between like a good temporal resolution, but also gives you like a good spatial resolution because you can study the cortical regions, but also the subcortical regions in the monkey. All the studies I show you after were done in macaque monkeys in the lab, and the fMRI studies of monkeys were quite developed over the last 10 to 15 years in different labs of the world. So at the beginning, one thing that you could told me that is if you used fMRI, it could only be like you study a vascular signal and not at all the neural signal of the brain. I think one good paper of the bold signal of MRI was done by Logothetis like 15 years ago, showing that when you have an fMRI signal, this study was done in monkeys. He showed the monkeys visual stimuli. He could activate the visual cortex in the monkeys using fMRI, and in the same time he did in this monkeys electrophysiologic invasive studies showing that this signal is not only a vascular signal, but it's really an electrophysiologic signal, and that you have a neural activation of the monkey brain when you present some visual stimuli, what was done with local field potentials in these monkeys. So when you do fMRI studies or functional imaging studies, there are two different techniques we can use. The two different techniques is that you present a stimulus to your monkey or to your human, which could be like an auditory stimuli. I will show you some studies using auditory stimulations using fMRI, and after you do your acquisitions, your subject is doing his task, and after you have a lot of data processing after to get this activation maps, like you see your brain, and what you see after in red or yellow is activations you get when you're doing one specific task. That was like the classical fMRI studies described in the past, and when first fMRI was used, it was like a stimulus-driven fMRI. What is more new technique using fMRI is what we call resting state fMRI. Resting state fMRI is that your subject has to do no task at all. So he's inside the scanner, he's doing no task. Often you ask him to have his eyes closed, and after, with a lot of data processing, after you acquired your data, you look what is functional correlations between different regions of the brain. So often you do the parcellation of your brain, and you want to know how the region, for example, the yellow one is interacting with the blue regions, and how are the spontaneous fluctuations of the brain in these different regions. So the mechanisms classically described for general anesthesia is that during anesthesia you have like disruption of cortical information integration, and with disturbance between the balance of feed-forward and feedback information in the brain. So the feed-forward information is that you have like an incoming stimuli, like for example an auditory stimuli. It's coming to the brain, it goes up to a whole network in the brain, and after the feedback is like it goes down, it goes back to the initial part where the stimulus happened, and it's this part, like the feedback of the information, that could give you the possibility to have an integration of the information you received at the beginning. So in the wake state, you have like a balance between the feedback and the feed forward, and during general anesthesia, classically, the feed forward is preserved, but you have like a breakdown of the feedback of the information in the brain. So the experiments we did in the lab are done in macaque monkeys. So it's a primate model to study different auditory stimuli in the wake state, but also after the study the same stimuli under anesthesia, and to see how the information processing is modified under anesthesia. So to better describe the neural mechanisms of general anesthesia. So first, that's like the setup we use in the lab. I will first to describe this setup with a very simple auditory stimulation we used in the lab. So there is a wake part, so the wake setup for the monkey. There's the anesthesia part for the anesthesia setup of the monkey. So first, for the awake part, of course, you have to train the macaque monkeys, because if you have monkeys and at the beginning they doesn't want to sit in a special chair to do the task you want them to do. So they are implanted with an MRI compatible head post, and after they are sitting in a chair, so they could completely free moving their body in the chair, but like they have to do a task in this part. It's like they had a visual point to fixate. They were rewarded when they were fixating this point, and they were passively listening to auditory stimulations that we presented to them. So first, we've presented very basic auditory stimulations to see only if we could, in the wake state and anesthesia, activate the cerebral cortex. So this were low-pitched and high-pitched sounds, like this one. And then, under anesthesia, it was the same monkeys who did the experiments in the wake state, but also under anesthesia. For the anesthesia, it could be propofol anesthesia to have an enhancement of the GABA receptors in the brain, or ketamine anesthesia to have an inhibition of the NMDA receptors in the brain. So the monkey received either one anesthetics or the other anesthetic. They were intubated, ventilated, because they were not breathing during the experiment, and also to have a very stable CO2 level in the blood, to have a very stable saturation in the blood. The body temperature also was very stable, because that things that's very important for fMRI studies, because if you change your CO2 level, you change your temperature, then at the end you cannot at all interpret your fMRI results. So they had anesthesia. The anesthesia pump was outside the scanner. So everything that's like in red is inside the scanner room, was a 3 Tesla MRI scanner. Everything that's blue is outside the scanner room. So they had anesthesia. They were inside the scanner, where we see the scanner there, intubated, ventilated. They had an EEG to monitor the anesthesia level, to make sure that during the whole experiment they were under the same level of anesthesia. They had in yellow their headphones to listen to the paradigm we present to them. And they had the whole physiological monitoring with heart rate, CO2 level, saturation, blood pressure. And all these signals were recorded outside the scanner room to make sure that you can use them after in your fMRI analysis. So also we make sure that they have the same level of arousal during the acquisitions. So we used what we call the monkey sedation scale, where we could define 4 different levels. We mainly used general anesthesia, but also for propofol we used what we called moderate sedation. So if you are in the wake state, you are alert. If you present juice to the monkey, of course he will drink, he will move spontaneously. If you are doing shaking of the monkey, he will move. If you do a 2-pinch, he will withdraw his foot, and he has a corner reflex that is present. So that's for the wake state. Under general anesthesia, you will lose all these spontaneous movements, and also if you shake him or do a 2-pinch, he will not react at all, and he has no corner reflex that's present. For propofol, we did 2 levels, also like a moderate sedation level, where then when we did shaking or 2-pinch of the monkey, he could have a clinical reaction, but spontaneously there was no movement at all of the monkey. And the EEG was monitoring these levels of anesthesia, like with the wake states, you have like posterior alpha waves on your EEG. When you go to a propofol anesthesia, you have first like theta waves that appears on your EEG, and when you go deeper with propofol for general anesthesia, what is typically described as you have very slow delta waves on your EEG. But propofol is very special because in the frontal region, this gives you like a peak of alpha waves around 10 Hertz. in your cortex. So you can make sure that your anesthesia level is the same during the whole acquisitions. And with ketamine is quite different because it gives you like rapid beta waves in the cortex, but also underlying delta waves on the cortex. So that's for the monitoring of this data. And so when we first present this very simple auditory stimuli to the monkey, so it was a sound, and we compared this sound to the baseline where there was no sound in the wake state under moderate and deep propofol anesthesia and under general anesthesia of ketamine, we could activate the auditory cortex in the monkeys for all the stimuli, but also for low frequencies and high frequencies stimuli. So it was possible to have a model in the monkeys that could be used to study auditory fMRI in the wake state and an anesthesia, because the auditory cortex in the wake state, but also anesthesia, remains receptive to the incoming sensory stimuli. So, but that's not only what we wanted to know, it was only to establish the model we will use after for our studies, because after we wanted to use a paradigm that's much more complex. It's an event-related auditory paradigm, what we call the local global auditory paradigm, first described in humans and then what was used in monkeys. So I will describe you in detail this paradigm because all the studies I will show you after is using this auditory paradigm. So this auditory paradigm is based on sounds. There are 5 sounds. These 5 sounds could be 5 identical sounds or 4 identical sounds with 1 different sound, what we called 1 deviant sound. Sorry. So if you are listening to, I hope that it will work. That's for the 5 identical sounds. Sounds were presented during 500 milliseconds. Or it could be this kind of sounds represent to the monkey. So at first you want that your monkey learns like a rule. This rule could be either 5 identical sounds or 4 identical sounds with 1 deviant sound. When you have only on the trial part a deviant sound, we will call it a local deviant because it appears in these very short sequences of sounds. And then, like, he has to learn this rule. You have like 5 identical sounds and so on. And at some moment, like in 20% of the presentation of the stimuli, you get like the opposite part of the paradigm, like 4 identical sound and 1 deviant sound. And in this part of the paradigm, this is what we call after like the global deviant. Because it's unexpected to get this when you learn that the rule will be like 5 identical sounds, and so on. And also we did it the other way around, like you hear all the time 4 identical sound, 1 deviant sound, and so on. And at some time, you will have in random order 5 identical sounds. So when you hear this, it's like really unexpected because you are expecting to hear all the time the rule you learned at the beginning, and you get an unexpected sequence, what we will call the global deviant. The study before it was done in monkeys was done in the lab I work in humans. In humans, it was done using event-related potentials, but also fMRI in the same humans. When you do event-related potentials in these humans, and you get like your deviant sound, like you have 4 identical sounds, you get 1 different sound, it's a local deviant, like a first order of perception that it's something wrong what you expected. On the ERP, it gives you what you call the mismatch negativity, and what you look to what corresponds this in fMRI studies give you an activation of the auditory pathway. What you see there is the auditory cortex, but you can activate the whole auditory pathway. So the detection of this unexpected sound, it's happening in the auditory pathway. But what's more interesting, it's like these second-order deviance you perceive in your brain, like you are learning a sequence like 5 identical sounds and so on. At some moment you get 4 identical sounds with 1 different sound, or the other way around. Then on the ERP you get a positive wave about 300 milliseconds, what is called the P3B. And when you look what happens in the fMRI studies, you not only activate your auditory cortex, but you will activate a whole network. And this whole network is not only the auditory cortex, but it's in the frontal cortex, in the parietal cortex, the temporal cortex. And what was shown in this paper, you have really to be conscious to activate this whole auditory pathway. Because if you distract the humans, like you are showing them other visual stimuli in the same time, like they are listening to this auditory stimuli, then you don't get this activation at all. At Tristan Beckenstein, Lynn, Lakash, and Stanislas Dehaene, who did the study at the beginning, after also did studies in coma patients, and what they could show that if you use this same paradigm in a patient that is in a vegetative state, he will not at all get this global activation of the brain. So it was like a marker established, at least in humans, for the perception, for the conscious perceptions of stimuli. And also this whole network that you activate, which comprises the prefrontal cortex, the parietal cortex, the temporal cortex, is what is described as the global neuronal network workspace, was a theory established by Stanislas Dehaene, which is director of the lab I work in. So it's what is a global neuronal workspace for those who are not familiar with this part, is that if you receive a sensory stimuli, like in this part, like the auditory stimuli received, first it arrives to the auditory cortex, but then to become really conscious to the brain, it has to be broadcasted all around the brain to get the information and the integration of the information. So once that it was described in the humans that this paradigm could be used as a marker of consciousness with this local global paradigm, we wanted to use it in the monkeys, because what was not sure at all if the monkeys could perceive the stimuli as the humans did. Though it were monkeys, the monkeys that you saw before, that were trained to sit in the fMRI scanner. The acquisitions were done, the monkey was passively listening to the paradigm. He has no instructions at all for the paradigm. And first we did the study in the wake state. So was a wake state, the monkey is sitting in his chair, he has his head blocked, he's listening to the stimuli that he received with MRI-compatible headphones. He is fixating a point to make sure that he will be quiet during the acquisitions, and he is rewarded during the acquisitions of the paradigm. So first, when you look at all the sounds compared to rest, of course you activate the auditory cortex and the whole auditory pathway. Of course, you have to make sure that first you activate the auditory pathway after that you look at more complex analysis of the stimuli. What is about the local novelty? So the local novelty is only that you get an unexpected sound in the trial you present to the monkey. And what we could show that, like the humans, this unexpected sound is perceived in the auditory pathway. There it's the auditory cortex of the monkey, that's the monkey brain that you see there. The activation is in red and yellow, and it's happening in the auditory cortex, but also like in the medial geniculate part of the body. So it's the whole auditory cortex that could perceive this first order violation of the brain. Before these fMRI studies, there were studies done in the monkey that showed in electrophysiological studies that the monkey could perceive like mismatch negativity in the auditory cortex. So it was not surprising that we could activate the whole auditory pathway with this first order violation detection in the monkey brain. But the question was, could he consciously receive this the stimulation with the detection of a global novelty in the brain. And the monkey activated the same pathway that the humans did in the brain, like it was in the auditory cortex, but not only in the auditory cortex. So for the global violation, so what is detection of a different sequence in the sequences that he learned as being the rule of the paradigm, he could activate the prefrontal cortex, the parietal cortex, which is there in the monkey, and the auditory cortex. And when you are doing plots in the fMRI studies for all these regions, so this represents the signal change of the MRI sequence. What is frequent is what is the rule the monkey learns. So either it's 5 identical stimuli in blue, and the deviant stimuli would be 4 identical stimuli with 1 different sound, or the rule the other way around. You all the time perceive that the signal is going up when we receiving this deviant sequence in the brain. So the monkey was, as a human, capable of detecting global novelty in the brain. So what was quite good for the studies after that, because we wanted to use this model for the anesthesia studies we will do after. Also, we did some.
What's the monkey supposed to do?
The monkey had no task for the auditory stimulations. The monkey was sitting inside the scanner. He was looking at a red dot. He was rewarded when he was looking at the dot, only to make sure that he was quiet in the scanner. Yeah, but he had no response to do at all. The humans at the beginning of the study, when they did it for the PNAS paper, were asked to count when they were detecting a global deviant, but after we did the same study in the humans at Neurospin, what was not published, to make sure if the humans were only passively listening to the stimuli with no instructions at all, that you could activate this prefrontal parietal network, and the humans were capable of doing it. So
that means we cannot really be sure that for the monkey this is affecting the state of consciousness, because we have no reportability. We have
No, they are not. They are not reporting it. But what was shown in the humans, when they were not sure to report the stimuli, they were not activating this frontal parietal network. So at least if you do an extrapolation to the monkeys, was like,
sure,
but you can, no, they were doing no task at all. Why it was done like this? Because after we want to compare what happens under anesthesia. And the idea was, if you train the monkey to do the task, then you don't know what happens under anesthesia. If it is when you see changes of this network, if it is only because he is doing no task, or because he's receiving anesthesia. So it could be, but we never did a study to ask the monkeys to report when they are doing the task. Could be possible, but it already to train the monkeys to do awake fMRI studies with passively listening to stimuli, it takes a few months only to get them trained to stay quiet in the scanner. But it could be.
No, look, it's all fine. Just
to make sure. To make sure that.
This by inference, right? So
yes.
You know what happens in humans, you see a similar pattern in monkeys,
and then,
So let's assume, given this compatibility.
Yes, that's at least the assumptions we made. Right. But they're doing no task for the paradigm. Yeah, that is sure. So we did also some other analysis of this fMRI study, not only using the classical contrast like you put for the global effect, like you put this rare stimuli minus this frequent stimuli, but doing a signal error propagation across the brain for this global effect, like you are looking what happens to the fMRI signal in the auditory cortex, and what happens to the fMRI signal inside this global novelty detection, and you get the activation of the same pathway than for the simple global effect contrast with really the signal that's doing the same behavior in the prefrontal cortex, parietal cortex, and posterior cingulate cortex. So what we described, or at least what we conclude in this paper, that like the humans, the monkey had like a macaque global neural workspace in the same state using this stimuli. And that the macaque, like the humans, were capable of a hierarchical proceeding of information in the brain through this macaque global neural workspace comprising made of the frontal cortex, the parietal cortex, and cingulate cortex, so the anterior cingulate cortex and the posterior cingulate cortex. And this was reading with a striatal homologue with the humans in the brain. So this local global paradigm that I described to you in detail could be used as at least a marker for consciousness perception and predictive coding in the monkey macaque brain. So that's for the wake state. So what we wanted to know, what happens under anesthesia, because one thing that could happen that you have like a completely shut down of the brain, you see no activation at all. But I will show you that's not what happens under anesthesia. So the two anesthetics we used in the monkey was Propofol to have one anesthetic agents that will enhance the GABA receptors in the brain, and one other, the other anesthetics used was ketamine to have an inhibition of the NMDA receptors, because there's a theory about that the feedback in the brain is based on NMDA receptors. So if you block your NMDA receptors, normally you should block the feedback in the brain, and so normally you should get at least no global effect at all in the brain, at least with ketamine. So the first questions of all, of course, was if the primary sensory network persists, like if you get auditory activations, but we were quite sure that we would get them. But then what happens in particular to this second order detection violations, like the global effect in the brain, and the question was if the monkey brain was still capable of a perception of cognitive function under anesthesia.
Lynn, before you go there, you said, you emphasized that you expect an effect, especially with ketamine, you said.
Yes,
so why do you expect a stronger effect with ketamine than a propofol in this case?
Why? Because at the beginning we took these two different drugs because propofol enhances the GABA receptors in the brain. At least it's not a pure drug because it's impossible to have pure clinical drugs only to one receptors. And ketamine blocks the NMDA receptors. And the idea is, at least in the theory of the global neuronal workspace, that the feedback is based on NMDA receptors. So if you block your NMDA receptors, normally you will have no feedback at all in your brain. And because the idea is that the global neuronal workspace is working on the feedback, so if you block your feedback with ketamine, you will have no global response at all in the brain.
But ketamine acts mainly on the NMDA receptors on GABAergic cells.
Yes.
So are you saying that the top-down feedback in Cortex is largely capitalizing on the GABAergic network?
Yes, could be, but
No, but there are interesting implications, right? That's sort of as an excited
Yes. That at least was the idea why we used two different drugs with two different molecular targets. Of course, it would be better if you have really pure drugs that only act like on one receptor, but it's quite difficult to get it. Yes, but at least that was the idea of the project at the beginning. So for the experimental setup was like the same I described you before. Like the monkey is in your scanner, the 3 Tesla MRI scanner. He is passively listening to your stimuli. He has the EEG on his head. He is intubated, ventilated, monitoring of physiological parameters, and the anesthesia is stable during the whole acquisitions. And to monitor the anesthesia level, the monkey had EEG. So also he had an evaluation of his arousal level at the beginning, at the end of the sessions. And then we did like a general anesthesia ketamine anesthesia. So because ketamine could be used at sub-anesthetic doses, but we didn't do it in the study. It really was like studying general anesthesia. To compare, we had general anesthesia with propofol and a more intermediate level for moderate propofol anesthesia. So first, when you put all your auditory stimuli together, you will see that you have an activation of the auditory pathway, either under propofol, moderate or deep, and ketamine anesthesia, with what is in yellow, it's auditory cortex of the monkeys, whatever anesthetic you use. When you look at the local novelty detection in the monkey, under propofol anesthesia, there was no detection of local novelty detection at all in the brain, but what is in agreement with the literature, because it's what is shown in the literature that if you use propofol anesthesia, at least in electrophysiological studies in humans, you will lose very rapidly the mismatch negativity in the brain. And with ketamine anesthesia, normally, if you can increase your level of ketamine anesthesia, the mismatch negativity goes down in the amplitude of electrophysiological recordings. And what we could show in the monkey that we have no detection of this local effect in the auditory pathway, but in some other regions that were described before, like subcortical regions of the striatum and the thalamus, this local deviant detection was still possible, what could be explained by the fact that ketamine is acting mainly on the cortex and not up subcortical regions. These regions were the same regions that also were activated for the local effect in the wake state. But and when you compare the wake state to the anesthetic states for the local deviant, so the detection of this deviant sound in the trial, it was all the time a detection that was stronger in the auditory pathway when you compare every time the wake state to the anesthetic state. But the more interesting part is probably this one for the global effect, what happens under anesthesia. So for ketamine anesthesia, there was no detection at all for this global deviant in the brain. But this was not the case for propofol anesthesia. If you take moderate or deep propofol anesthesia, you could still detect this global effect in the auditory cortex of the monkeys, but also in some prefrontal regions of the monkeys, either under moderate or deep propofol anesthesia. What was quite interesting, because if you go back to very old theories about anesthesia, it was like you have a shutdown of the brain, and it's not at all the case that you have a shutdown at all, a complete shutdown of the brain under propofol anesthesia. And at least for this anesthetics, for propofol, you have still a perception of a cognitive function of your brain. But if you take the whole network, like the prefrontal parietal temporal network that was described as being the macaque global neural workspace, what you block all the time, you have never detection of the global effect in the parietal cortex, and you have never detection in the posterior cingulate cortex. So some information could go from the auditory cortex to prefrontal regions, but it never goes to the parietal cortex. So there seems to be like a shutdown of the detection of information in the parietal cortex.
Can you indicate this more precisely? So where do we see exactly that the parietal cortex is really disabled?
It was disabled if you compare after your wake state, because we took it was the same monkeys that were scanned in the wake state and under the anesthetic conditions. And we did a comparison of the wake state and all the anesthetics. And then you see what is every time stronger activated in the wake state. The prefrontal regions were stronger activated, but some other prefrontal regions were activated under anesthesia. But all the time, what was really consistent with all the data were that you find this IPS, what is the interparietal sulcus of the parietal cortex of the monkey, was really, really strong activated, much stronger activated in the wake state. And when you look at all the fMRI data, you never find, even if you plot the data in the parietal cortex, you never had a global effect in the parietal cortex of the monkey. So whatever the anesthetic use, but also for propofol, even if propofol could get activations in the prefrontal cortex. So also it was the case for some specific nuclei of the thalamus, like the parafascicular nuclei of the thalamus. What Bashir will show you later why we use some specific targeting of this nuclei to stimulate. But if you look only at the cortex, it's like the parietal, you have a complete shutdown of the information processing in anesthesia in this parietal cortex. Even if you have differences between, and when you compare the differences between the propofol and the ketamine anesthesia, you see that propofol could activate the prefrontal cortex, but not other regions of the brain. So to conclude, at least for this local global paradigm used in the monkeys in the wake state and an anesthesia, it's that for the wake state you get a local effect. It's like the detection of these one deviant signs, mainly in the auditory pathway. The global effect represents like the global neuronal workspace of the macaque brain, which is happening in the prefrontal cortex, parietal cortex, and auditory cortex of the monkey. When we go under anesthesia for propofol and for ketamine, you are still that your primary sensory cortexes, like the auditory cortex, is still active under anesthesia. For mainly propofol, you have a persistence of a cognitive function under anesthesia, but you have a complete disorganization of the global neuronal workspace of the monkey, and with a missing piece that is common for all the anesthetics is the activation in the parietal cortex. But also we could demonstrate that you have like a specific network for the wake state with robust activations in the parietal cortex, prefrontal cortex, and in some thalamin nuclei like the parafascicular nucleus of the thalamus. So that's when you want to study information processing in the monkey brain under anesthesia. But it's not the only studies we did in these monkeys, because also we wanted to see what happens at the brain at rest, because if you go to clinical anesthesia in everyday life, you will not present stimulations to your patients, but it's mainly that the brain is at rest during under anesthesia. And when you study when we wanted to study the brain at rest, we wanted to compare the function of the brain, like to the structure of the brain of these monkeys. So the resting state studies were first done in the wake monkeys and under propofol anesthesia with 2 levels of propofol anesthesia, like moderate propofol anesthesia and really a deep general anesthesia of propofol. The setup is the same that was described before for the auditory stimuli. And so first we did a parcellation of the monkey cortex. So we only looked at the cortical regions of the monkey. We didn't do for the moment studies of the profound regions of the monkey, like the thalamus or like the striatum of the monkey. So it could be done in a future step, but isn't done for the moment. So if you took one hemisphere of the monkeys, there was done a parcellation using the Cocomac atlas of the monkey, and you get like 41 regions on every hemisphere of the monkeys that's represented in different colors here on the monkey brain. And we first look at stationary functional correlations in the monkey brain. So you will take the whole data that you acquired at resting state. The resting state was acquired during 20 minutes, and you do an average of the connectivity that you get in the monkey brain for this 20 minutes of acquisitions in the monkeys that were either in wake state or under propofol anesthesia. These correlation matrices are represented here. So like every line represents one region of the monkey brain, like for the right hemisphere, for the left hemisphere. And when it's more red, it's like you have high positive correlations. When it is blue, you have high negative correlations, and it is like green, you have no correlations at all in these regions. So you see that you have in the wake state a lot of positive correlations, a lot of negative correlations. And when you plot these to glass brains of the monkeys, like in red as a positive correlations, in blue as a negative correlations, you see that even at rest, when there is no task at all that the monkey is doing, because the monkey is sitting in the dark in the scanner and he has had no task at all that he was doing, that you have like the brain is highly connected at rest, at least in the wake state. When you go, when you apply anesthesia in these monkeys, what you see first, that you have a lot of correlations that persist in the brain, but which seems to be mainly positive correlations of the brain. So when you lose consciousness, it seems that you lose these negative correlations of the brain, and it was like one idea that negative correlations could be information processing of the brain, so you lose them under anesthesia. And this is like you have the positive correlations are very high in the monkey brain in the wake state, so this represents the wake state. This represents moderate propofol anesthesia, deep propofol general anesthesia, and if you do the ratio of the negative to the positive correlations, they are much higher in the wake states than under anesthesia, which are significantly different under anesthesia compared to the wake state. But it's not only the only analysis that we did in these monkeys. So what we did also was dynamic correlations analysis of the monkey, because if you are doing nothing during 20 minutes, it was shown first in humans by the study of Lynn, that the brain is not in the same state during the 20 minutes where we are in a resting state. The state of the brain could change, because at some moment you could think about something, and some other moment, perhaps doing a task or so on. So when you look at this 20 minutes of acquisition of resting state, it's like the connectivity of the brain is changing. So if you do only stationary functional connectivity analysis of the brain, you get like one state, but perhaps it's a state that could be even impossible in the real life, because it's an average of 20 minutes of acquisition of a signal. So it would be more interesting to look at these dynamical correlations of the brain, like you do a Kamin's grasping of your data, like you put all your data together. In this case, it was the wake monkey data, the moderate propofol sedation, the deep propofol sedation. And also what we wanted to do is to compare them to the underlying structural connectivity of the monkey. So you get, we fixed the number of states at 7, because that was done before in the human studies that were done. So you want to know if you can get 7 clusters in your data, and then we compare the 7 clusters to the underlying structural matrix of the monkey. The structural anatomical connectivity of the monkey was not acquired in the same monkey. It was used from tracer studies with COCOMAC database of the monkey. So you had your 82 regions of the brain, and you get 7 states. When you look at the 7 states, you can get them in the wake state. So your monkey could be in whatever state during the 20 minutes of acquisition of the data. With some states, like the state 1, that is, if you see, you have a lot of positive correlations of the brain, but also a lot of negative correlations of the brain. If you look at state 7 is like a state that is not highly connected in the brain, but it's a state that it's closest to the underlying anatomical connectivity. So the data were sorted like state 1 is the one that is the least, is not closed at all to the underlying anatomical connectivity, and state 7 is the one that is closest to the underlying anatomical connectivity of the monkey. So in the wake state, you get like the 7 states that could be present in your data. If you look at the probability of appearance, it's like the state even 1, 2, 3 as the one that are the most probably, and the state 7 seems to be the less probably in the acquisition of this monkey data. And when you go to anesthesia, so you have a loss of consciousness, what you will see that under deep general anesthesia, like the state 1, 2, and 3 never appear in this data. So these states that are where you have a lot of positive correlations, but also very much negative correlations, you never get them under anesthesia. And even if what it's predominant under anesthesia is the brain state 7, so the state that is the closest to the underlying anatomical connectivity of the monkey brain. But the brain is not in one state under anesthesia. It could still go to switch between like state 4, 5, 6, and 7. But these states 1, 2, and 3 never appear in the monkey brain. So the networks defined by structural connectivity present during propofol anesthesia, and they are quite different for the one of the wake state. We did some entropy measures of these brain states. When you do entropy measures, you see that for the state 1, it's a brain state with a very high entropy, and that for brain state 7, it's a very low entropy measure of this state. So that's for propofol anesthesia, but I told you that propofol is acting on GABA receptors on the brain. So we wanted to know if we can generalize this message to other anesthetics, because, okay, it's nice you have this data with propofol, but is it only working with the wake state and propofol, or could it work with other anesthetics that we test, like for ketamine anesthesia, but also we want to test another one, what is sevoflurane anesthesia, to really generalize this to all the anesthetics that are used in clinical practice today. So the same monkeys were scanned also with ketamine anesthesia with sevoflurane anesthesia, first for stationary functional connectivity, and you get similar results to the propofol anesthesia, like you decrease your stationary functional connectivity with the positive correlation, the negative correlation. And when you do the ratio of negative to positive correlations, it goes down under anesthesia. Even it seems to be less under sevoflurane anesthesia than under ketamine anesthesia. When this is a comparison of these statistical maps and what is significantly different between the awake data and the anesthesia data are the p-value values that are in blue, and you see that you have a lot of differences between the wake state and the anesthesia state. But also what we wanted to know in another part in this data are not published for the moment, that if you look at the keynotes of the global neuronal workspace, like we defined them in the wake monkey studies, if you are presenting no paradigm at all to these monkeys and only look at these regions that were identified on the data before, what happens to these regions at rest? So these are the correlation matrices of the wake state. Then you have the moderate propofol, the deep propofol, then the ketamine anesthesia, and the 2 levels of sevoflurane anesthesia. It's like one region that you see there. It's prefrontal cortex, like frontal eye field of the monkey. You have like parietal cortex of the monkey and posterior cingulate cortex of the monkey. In total, we described about 10 regions of this. And what you see that if you do a schematic representation of the correlations of these regions at rest, you see that even at rest, these regions are highly correlated, either positively or negatively, in the brain, but that under anesthesia, you have a lot of correlations between the global neuronal workspace nodes during anesthesia in these monkeys, what is schematically represented, even if your monkey is doing no task at all. So we did also this dynamic functional correlations analysis in the monkey under ketamine and after under sevoflurane anesthesia, and you get like a similar profile, whatever anesthetics you will use. That's the state 1, 2, and 3. We never presented under anesthesia. So if your monkey is not present, it will never be present. And under anesthesia, the networks defined are the one that is close to the underlying structural connectivity of the monkey. You see like the same data for sevoflurane anesthesia. In the wake state, the 7 states are present, and when you lose consciousness, the 7 states go down to 3 or 4 states in the monkeys. And when you look also in the keynotes of the global neuronal workspace in the monkey for the dynamic functional correlations, you see that in state 1, all these keynotes of the global neuronal workspace, like prefrontal parietal cortex or posterior cingulate cortex, are highly connected in the monkey brain. So which represents state 1 in the monkey brain. And when you go to state 7, it's like these keynotes of the GNW are not connected at all, really, in the monkey brain. There's some connections that persist in the prefrontal cortex, but they're never connected like to parietal regions or to posterior cingulate cortical regions. So what could we conclude from these resting state studies? That in the wake state, the brain has a lot of functional connections, correlations in the brain, which are either positive or negative, that the brain could have at least 7 states that could be defined, that these 7 states are states that are close to the anatomical connectivity, but the brain can go to states that are not at all close to the underlying anatomical connectivity. And that under anesthesia, the brain for stationary functional correlations is weaker connected, with a lot of loss of negative correlations in the brain, and that for dynamic correlations analysis, you get less states compared to the wake state, and that is the states you will lose as the one that are the farthest away from the underlying anatomical connectivity of the brain. So like brain state 1 with high functional correlations within the global neuronal workspace is completely lost under anesthesia, and that brain state 7 with very low functional correlations in the global neuronal workspace is present, and big pumps really dominate under anesthesia, and that the networks present under anesthesia are defined by the structural underlying connectivity of the monkey. So when we go to the general conclusion of this talk, that what we could show in the studies we did in the lab is that the auditory cortex is still receptive to incoming sensory stimuli in the monkey, so that it seems that the feed forward is present in the monkey brain, that you have a reduction of the repertoires of spontaneous brain activity in the brain, with an increase of brain states that are very similar to the underlying anatomical structures in the brain, that the anesthetics disorganize the global neuronal workspace in the monkey brain, but in some part there's some cognitive processing still present under anesthesia, and that you find very low functional correlations within the global neuronal workspace in the monkey brain, and that at the end you have a loss of consciousness of your monkey. So could there be some perspectives of this work? Of course, you have to generalize this to all the anesthetics that exist, in particular to dexmedetomidine studies we didn't do for the moment, because it has a completely different action in the brain compared to propofol, ketamine, or sevoflurane anesthesia. And then if you want to really make sure that these regions, like the parietal cortex, would be regions that are very important for the conscious processing of the brain, you have to probably to do like some pharmacological inhibitions of these regions, like perhaps using mucimol to inhibit these regions, and then even in the wake brain you could perhaps have no conscious processing in the brain. And also the studies could be important to develop in the future better monitoring of anesthesia in the operating room, because today there exist some devices, like for the one that familiar perhaps, like the bispectral index, based on monitoring of the depth of anesthesia, but I think clinicians would need better monitoring than this one. And also the idea is that in the future perhaps you can get like a closed loop anesthesia. You have like a monitoring of your brain, then it's connected to a pump, and the pump is administrating the anesthetic you use, and not interfering with the anesthesiologist that is in the room, to get to be sure that your patients receive really general anesthesia during the whole process of the anesthetic procedure. And also, the idea is to use this model to study recovery of consciousness in the monkey brain, with this talk that will take place later with Bishir Raya, based on these studies. Thank you.
Thank you, Lynn, for this very interesting and informative talk. So we open the floor for questions. We have a question here.
Can you please explain why is state 7 in dynamic functional correlations the closest to anatomical connectivity?
It was like statistical analysis we did when you compare the correlations present, and if you do clustering, that's the one that's the most closest, because it's the one that has nearly only positive correlations in the brain. There are no negative correlations at all. And in atomic, when you look to anatomical connectivity, you have like only positive correlations. And if you compare the regions of the monkey brain that are connected, as the one that are the most connected during also the anatomical tracer studies in this monkey. So if you go to brain state 1, when you do the comparison of this state, you have a lot of anticorrelations, and anticorrelations are not in the regions that you have the anatomical connectivity.
Hi, just following on what he asked. What do you mean by correlations in structural connectivity?
No, you have no correlations. Okay, I'm okay. Yeah, you have it. The anatomical connectivity was not done in the same monkeys. It was done on tracer studies, so it's based on atlas studies that are published. So it was, you have like brain region X and Y, and they looked if there was an anatomical connectivity between these regions. And in our studies, we compare the functional correlations that are present, the regions where you have like a positive correlations or negative correlations, and how these regions like X and Y match to X and Y of the anatomical connectivity.
Okay. Another question is, why did you do this study on monkeys?
Why on monkeys? One reason is why not? I don't know if your question is why not doing them in humans.
Yes, why not in humans?
Yeah.
Because
yes, why not doing them in humans? You need a lot of data to do the analysis. And often, if you go to the operating room, because one question is why not doing them in the operating room with patients that are having a general anesthesia, often you are not using, if you're doing a general anesthesia to a human, you use hypnotic agents like propofol, ketamine, or whatever to induce loss of consciousness. But during the anesthesia in the operating room, yeah, also using like other drugs like opioids or even other drugs to maintain blood pressure stable, and it's very difficult to study the whole process of anesthesia during a surgical procedure. And also you need a lot of data to do the analysis.
But can't you just give only the anesthetic thing to the person and to do the study?
So to take human volunteers and do it?
Yes.
Yeah, but you have to find them. I think it would be very...
Young people love this stuff.
Can't you pay them?
Of course, it could be possible. I think anesthesia is quite safe today, but going to a general anesthesia level in humans, it could be quite tricky because you have to intubate your patients and mechanically ventilate them to go really to a general anesthesia level and to make sure that it's completely safe. Anesthesia is quite safe, but you never can guarantee that it's 100% safe, and there will not be a problem with the human. And if you take a healthy volunteer and only want to study anesthesia, it could be tricky. I think you have to find...
Are the effects in monkeys the same as the effects in humans?
Yes.
She just said if we wanted some ketamine. I remember that ketamine was used as a recreational drug. But I don't know if it's the same ketamine.
Yeah, but it's not ketamine used as sub-anesthetic doses. It could be recreative if you use it as sub-anesthetic doses, but not if you use it at general anesthesia level doses.
Depends what kind of entertainment you're in, right?
Yeah.
So there are not psychic effects on.
At least we tested the same monkeys with the local global paradigm after we did all the anesthesia studies, and they were capable to do this paradigm again. And animals said that they're really for several years in the lab and doing other studies, and they think are good, in good shape.
Okay, so Lynn, thank you. This is fantastic work, right? And it's really a tour de force. It's really complex to run this and to analyze it. On the other hand, as always, there's surprises, right? And
Yes.
one surprise is that if you look at the actual mechanisms of action of propofol and ketamine,
Yes.
they are acting in opposite ways on
the
your
brain.
inhibitory system,
Yes.
right? Ketamine
Yeah.
is taking it out of the game, propofol is enhancing its role,
Yes.
right?
Yes.
But now, if you, in the end, look at the impact on your global workspace, the very last experiments you were showing, it actually appears that the net effect is the same,
Yes.
right?
Yes.
There's an
Yeah.
overall reduction
Yeah.
of the correlations in the network.
Yes.
So that's rather surprising, right? So how do you explain that?
It's a good question how to explain it. You
Because in
could
some sense
explain.
it shouldn't make any sense, right? It doesn't make any sense.
It makes sense because all of them induce a loss of consciousness, even if they work at different receptors of the brain. So you have to find some reason why they all induce a loss of consciousness. But it could be surprising.
Like you say, there's a reduction of negative correlation.
Yes.
Well, negative correlation you would expect depends on inhibitory interactions
Yes.
across your nodes.
Yeah.
Well, that should be then enhanced with propofol and reduced in ketamine,
Yes.
if we follow the mechanistic
Yes.
model.
Yes.
But that's not what you see.
No, that's not
So
what
that's
you
why
say.
it's not making sense, right? So given the
Yes.
premise, the
Yes.
result is inconsistent.
Yes, it's inconsistent
So how
with
do
this.
you square that circle?
It's a good
Don't
question.
look at him.
I'm-
Monsieur, you don't know. Are you going to answer that in your talk, Monsieur? Are you going to answer that?
Not specifically, but it's true that actually whatever happened with the paradigm.
I mean, the talk could have started with the resting state data because this is very consistent and that generalizes the anesthesia phenomena, whatever the anesthetic. And then when it comes to a task paradigm, then things starts to be tricky. Actually, one way to still square the circle, as you said, is that you never obtain with this local global paradigm under anesthesia, you never obtain the whole frontoparietal network. This kind of global macaque global neural workspace, and it looks like it's all or nothing. If you take pieces of this GNW, then the full consciousness process stops down. So even though you may keep pieces of it, it looks like if you take main parts, either prefrontal or parietal, you end up having a loss of consciousness because you lose this broadcasting effect and this ignition effect by the end.
Okay. So, but then I had a little follow-up, and then I guess we go to our coffee break. Because, but isn't there one issue that when you talk about the mechanism of action, we talk at very different timescales,
Yes.
right? These are
Yeah.
rapid, short time constant mechanisms
Yeah.
that you are assessing in a very, very, very indirect way with a lot of
Yes.
spatial-temporal
Yeah.
averaging.
Yeah.
So there's maybe some process in between that we haven't really fully understand
Yeah,
yet. So
yeah,
maybe
and
one
one of the ideas is to do in the same monkey invasive electrophysiological studies to study what really happens in the
Exactly.
smaller time space. But that's what's one of the what we'll
course.
do in the future.
But then the other thing is, okay, there's the notion of global workspace, right?
Yes.
Going back to Bernie
Yeah.
Bars, and it was very global initially.
Yeah. Yes.
But it seems the global workspace is getting incrementally more local. And essentially, we're now talking about the frontal parietal network.
Network. Yes.
Is that fair? Shall we
Yeah.
say maybe we should just start talking about the frontal parietal network and forget global workspace because it's not global.
No, it's not global.
you agree?
But there's also part. If you only look at the cortex, okay, you could think that's frontal parietal, but probably there is another part that it's probably main part of it is the thalamus. And if you only think it's frontal parietal, then it's not.
Then you see the
Yes.
problem, right?
Yes.
I think
Yeah.
we
Yeah.
have to start
Yeah.
to redefine
Yeah.
a little bit where
Yeah.
the boundaries now of
Yes.
the
Yeah.
global,
Well, yes. Yeah. 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 Yeah.
which you
Yeah.
clearly shown your
Yeah.
data.
Yes. Yeah. 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 That's true.
No, beautiful.
One more question and then coffee break.
Okay.
I was just thinking about what Paul suggested was a bit of a contradiction. Isn't that related to maybe the way you measure things? Because I noticed that sometimes you don't really show correlations, but covariances. You had Z
Yeah.
scores.
Yeah.
But in the end, probably you use the Pearson correlation or something like that. And the Pearson correlation normalizes. So in one case, you remove the correlations because you
Yeah.
remove the negative feedback. In the other case, you're basically suppressing the activity completely because of excessive feedback.
Yes.
And that's why you probably get this, what looks like a contradiction, but isn't really one, because you use a Pearson correlation at the end, but not in your heat maps.
Yes.
No?
Yeah. That could be a possibility. Yes. Yeah. That's good suggestion. Thank you.
So we now have a coffee break and we reconvene at 11 for the talk.
Okay. Thank you.
Thank you, Lynn.